Before I begin the talk, I would like to thank Professor Rajneesh for kind invitation. So as you can see, and as introduced by the chair, I'll be talking about integrating microbial electrochemical processes into some of the existing approaches to achieve environmental sustainability. And you can see that microbiology and electrochemistry basically are two keywords here. You have seen some of the electrochemical talks today. So, and most of you are probably from chemistry background. Is there anyone who is from biology background here in the audience? No. So I'll then
remotely connected.
Remotely, yeah. So I'll spend some time on introducing this topic because that's what I think is probably the goal of this particular session. And then I'll talk about a few examples of biotechnologies which involve the use of this integrated concept. So this is the overview of my talk. First, I'll talk about extracellular electron transfer processes and the microorganisms that basically follow these kind of unique metabolic processes in order to achieve their respiration. Based on that, what is microbial electrochemistry and what kind of microbial electrochemical technologies can be developed exploiting these kind of processes. And I'll focus on three processes that we are currently focusing on, which are based on microbial electrocatalysis. The first one will be on sewage sludge management, in particular for the production of biogas. As you know, it's a renewable energy source. And then one technology that we have been developing over the last 6 years, which is abbreviated as iHydromet. I'll speak about that later on. And then finally, I'll end with the CO2 utilization using these processes. So, because when electrochemistry comes into the picture, electron transfer is very important. Now, how this microbial electron transfer is linked to electrochemistry? So, to understand that, it is important that we first try to understand microbial electron transfer processes. So, as you know, the flow of electrons basically is very important to all metabolic processes in life forms. So, including higher eukaryotic micro organisms like humans also. So, in our case, how exactly is electron transfer is involved in respiration? The oxygen that we kind of take in is reduced at the membrane level by certain enzymes in order to complete the the respiratory processes. Now, in case of microorganisms also, basically they have the advantage of using different kinds of electron acceptors. So, in natural environments, they always try to use the electron acceptor with the highest reduction potential and the electron donor with the lowest reduction potential for maximizing their energy gain. Now, in our case, we don't have that liberty. We can just respire using oxygen, but microbes can fine tune their respiratory processes according to the electron acceptor that they can access in a particular environmental setting and so on. Now, just a simple example in bioenergetics or in energy metabolism, if you take into account glucose as a substrate or electron donor and oxygen as the electron acceptor, basically this is the common pair in aerobic respiration. So, this is something is done by most of the living beings, including humans. We follow aerobic respiration, but microorganisms can go beyond aerobic respiration. So, for example, here I introduce a bit different respiratory process, which is the anaerobic respiration. Now, when we talk about electron acceptors can be of soluble nature and insoluble nature. Oxygen is soluble. Now, what you see here, certain numbers, these are nothing but the reduction potentials under standard conditions, pH 7 and so on. And if you pair this with glucose, for example, it has about minus 0.41 volt reduction potential. The gap determines the energy gain for microorganisms, even for higher beings and so on. Now, in the case of anaerobic respiration, that means when oxygen is not available in certain environments, microbes have evolved to use different other kinds of electron acceptors like nitrates, sulfate, elemental sulfur, fumarate and so on in order to achieve respiration. That means in order to grow under oxygen depleted conditions. But there are certain environments where soluble electron acceptors are mostly absent. or sometimes microbes are unable to find soluble electron acceptors. And this is obviously a very challenging conditions to grow. But some microbes have evolved to even respire using insoluble electron acceptors like mineral oxides, primarily iron bearing mineral oxides or manganese bearing mineral oxides, as well as large organic substances. For example, if you dig deeper into the soil, you will have a lot of humic substances or humus that can also serve as electron acceptors. And even microbes, some microbes can respire using other microbial cells. That means the other microbial cell can act as the electron sink for them. Now these are certain unique things which obviously other organisms cannot do. Now in such cases, now this is about electron acceptor, but electron donors can also be soluble and insoluble. So for example, elemental sulfur or reduced minerals. Now here these are oxidized minerals, here these are reduced minerals. So microbes can also use electrons, that means the energy from reduced minerals. Now in order to access these insoluble electron donors and acceptors, obviously microbes need unique mechanisms. Oxygen or other electron acceptors, they can easily cross the membrane barrier, cellular membrane barrier. They can enter at the electron transport chain and get oxidized or reduced according to whether it is energy gain or energy production. Now in this case, this is not what we can do, because oxygen can easily enter our cells. So to achieve respiration or to use these insoluble electron donors, microbes have evolved unique respiratory processes which are basically referred to as extracellular electron transport. So microbes, when they oxidize glucose or any other substrate, they transfer these electrons, or they have to transfer this electron outside of the cells. Now this process is referred to as extracellular electron transport, because electron transport is happening outside of the cells. The reverse can also happen, electron gain can also happen from the reduced minerals or other cells and so on. So this is referred to as extracellular electron transport. Now these kind of processes are very common in natural settings. I have illustrated here a few representative processes where this kind of electrical interplay is very common. So for example, in the first case, here this is mineral, represents minerals, and these are microbial cells. So microbes can use these mineral oxides as electron acceptor to respire. Microbes can use mineral oxides as electron donor to grow. These mineral oxides can also act as an electron conductor. That means they can store charge, and when microbes get close to them or attached to them, basically they can either take the electrons or transfer the electrons. So they can serve as the conductor. And finally, these materials can also act as battery, because they are storing electrons, they are storing charge and so on. And when microbes get in touch with them, basically they can achieve a lot of respiratory processes which otherwise are not possible for these microbes. Now this is something is very common in natural settings. Now how about changing these natural conditions in laboratory or in an engineered environment? So for example, the best way to replace these insoluble electron acceptors or donors, in other words mineral oxide, is to use or replace them by electrodes or conductive materials. Now electrodes or conductive materials can basically play the same role, either electron donor, acceptor, or charge storage and so on. And these are the different kinds of electron acceptors, these are donors. So if you can, now you know the electrochemistry. So when you talk about electron acceptor, it is in electrochemical cell, it is an anode, and in the case of electron donor, it is a cathode. So basically if we replace these natural electron acceptors and donors by electrodes in an engineered environment, we can still allow the microbes to achieve the same kind of functionalities in terms of respiratory processes or in terms of different kinds of metabolic functionalities and so on. Now understanding these interactions between microbes and electrodes or conductive materials or even mineral oxides is nothing but microbial electrochemistry. So this field has evolved over the last 1.5 decades. So a lot of focus is on understanding these kind of interactions and also trying to solve some of the basic respiratory questions that are associated with when the microbes respire on mineral oxides and so on. Now obviously for this you need an electrochemical system, and this is something is normally used in the case of microbial electrochemical technologies. So this is just a summary of like how we can integrate microbes with this electrochemistry. So this is simple electrochemical system where you have anodes, cathodes separated by a membrane. It can be a cation exchange membrane or an anion exchange membrane. Substrate oxidation happens at the anode and substrate reduction. Now this can be different. Substrates can be of different types in electrochemistry also, and we have seen some examples earlier. Now instead of using this metallic catalyst or some inorganic catalyst and so on, basically if you use microbes to enable some of the substrate oxidation reactions at the anode, it becomes bioanode, and some substrate reduction reaction at the cathode, it becomes biocathode. The CO2 reduction example that I will be speaking about, that is basically enabled at the cathode side, because it's a CO2 reduction reaction where cathodes serve as the source of electrons, or it can be used to produce hydrogen and so on. Now by just combining microbes with the electrodes, basically similarly similar to the electrochemical systems, like in the case of fuel cells, you can produce electricity. In case of electrolysis cells, you can produce hydrogen by putting in extra energy. So similarly, this can be enabled using microorganisms and electrodes. You can produce electricity, it becomes microbial fuel cell, and if you allow some electrolysis reaction, it becomes microbial electrolysis cell. So that is something is enabled using this kind of combinations. Now the microorganisms which have these capabilities of respiring or using electrons from cathode, these are referred to as electrochemically active or electroactive microorganisms, or in other words EET capable microorganisms. That means extracellular electron transfer capable microorganisms. Now this is applicable to both natural as well as engineered environments. They play various important roles, for example in cycling of various elements as well as interspecies electron transfer. I will speak about these applications also, how we are exploiting these interspecies electron transfer processes in some of the biotechnologies, and they can be utilized to develop various biotechnologies, which I will be speaking about. Now they can be further classified into two groups depending upon the electron transport. The first one is exoelectrogens. That means when the electron transfer is outside of the cell, that is in outward direction to the anode, it is outward EET. Those are referred to as exoelectrogens. And the other way, inward EET from cathode or from reduced mineral to microbial cell, it is they are referred to as electrotrophs. Now probably you have heard about autotrophs, the heterotrophs. The classification is based on carbon utilization. Based on energy utilization, you have lithotrophs, you have organotrophs. So electrotrophs is one of the new addition to that description in microbiology or in general in metabolic processes, which is somewhat closely related to lithotrophy, mode of energy utilization and so on. Now just how when you grow them on electrode, for example here you see just a simple three electrode configuration electrochemical system, where this is counter electrode and this is a working electrode. Now what you see here, this is also carbon based electrodes, but you see a very nice brownish color growth on this particular or color structure here. So this brownish color is because of the microbial cells growing on the electrode. Now when they are able when they can grow on it electrode, when they don't find any other electron acceptor in the surrounding, that means in this case in the growth medium, we ensure that we provide only such conditions where they don't find any other electron acceptor than electrode. So they don't have choice. So only microbes which are capable of now respiring using extracellular electron transport, they will be able to go to the electrode and eventually form. So keep in mind, when I say respiration, it is always linked to growth. So when they respire means they grow, and in microbes when they grow they multiply, and eventually you will be able to see that growth on the electrode. So that is what you see here. Now how do you confirm? This is a scanning electron micrograph where you can see a lot of rod shaped cells growing in that particular biofilm. You have still probably seen these kind of pictures, but mostly for pathogenic microorganisms like E. coli and so on. But these are some of the very interesting microorganisms which can also grow on electrodes and so on. Now how do they achieve this extracellular electron transfer? So this process can happen either via direct or indirect processes. In direct, the physical contact between microorganisms and the electrode is very important, where they can attach to the electrode and use membrane cytochromes somebody had mentioned iron is one of the important component, in also in metallic, electrocatalysis and so on. So similarly, in case of cytochromes, iron is at the center. So the previous talk was on inspired catalysis. So that's how everything is kind of also linked. So here, when they attach to the electrode, the electron transfer can happen via cytochromes directly, or they can also form nanowires. Obviously, these are not very big wires, very small wires, but packed with a lot of cytochromes and also some other metallic like conductivity and so on. So debate is going on how exactly nanowires can transfer electrons and so on. And the other mechanism is indirect electron transfer. It can be via some mediator, redox mediators, or it can be via some energy carriers like hydrogen or some format like molecules and so on. Now this can be enabled or this can happen in different microorganisms. So directly it is more promising because you don't have to add anything in the system, and you can have better efficiencies as per your target process are concerned. But there are some advantages with the mediator electron transfer also. But the point is basically the electron transfer happens via either direct or indirect electron transfer processes. Now, how can you explore these processes for developing different applications? This is just an overview of different reaction possibilities that at the anode side. These are at the anode side where you can oxidize anything that you can think of, but that has to be microbially oxidized. So microbes, as you know, can oxidize almost all kinds of naturally available organic substances, and even artificially ones like plastic degradation or a lot of anthropogenic substrate that we are adding to nature. They are also they can also be oxidized by microorganisms. At the cathode side, obviously you have a possibility of enabling different reactions, starting from just oxygen reduction that will lead you to produce electricity, hydrogen evolution reaction, that is through electrolysis. Now in conventional water electrolysis, you have to put in a lot of energy to split water, but in microbial electrolysis cells, that energy can be brought down considerably, and you can link the hydrogen evolution with any wastewater or organic matter oxidation and so on. You don't have to use pure water for microbial electrolysis process, and more importantly CO2 and so reduction and so on. Now some of these reactions, in particular the ones highlighted in yellow color, these are spontaneous reactions. That means you don't have to add any external power, whereas the ones that are highlighted in other color, these are the ones which need external powers, which also includes hydrogen evolution reactions. Now using all these combinations, now you can think of different applications, starting with electricity or hydrogen generation. Now the field actually started with electricity production, but now the community knows that electricity production to basically for our regular is something very challenging, but you can definitely explore these kind of processes for hydrogen generation and a lot of other applications like wastewater treatment. I will be speaking about one application where we are working on wastewater treatment, recovering different kinds of resources, for example nitrogen, as well as CO2 reduction and bioremediation, biosensing, bioelectronics. I talked about nanowires. So some scientists are now working on producing these nanowires in bulk, so that you can use them for different appliances and so on. So that is how field is now evolving. It started with microbial fuel cells, electricity generation, but we have realized that large scale electricity production using this particular concept is not easy and it's challenging. So we are now moving towards other kinds of fields and so on. So these are the three applications or the processes that I'll discuss about. The first one is conductive material assisted anaerobic digestion. Second is about wastewater management, and third is about CO2 reduction. Now how many of you know about anaerobic digestion? Probably most of you, or at least biogas plants. So you have heard about biogas plants. So biogas plants, in terms of metabolic process is concerned, it's nothing but anaerobic digestion. Actually anaerobic digestion happens in biogas plants, which leads to the production of biogas, and it's not a single step process, it's a multi-step process involving the activity of more than 4 different groups of microorganisms. The final one is methanogens, which actually produces methane, and which is actually the energy molecule in biogas. The rest is impurities including CO2 and so on. So as far as anaerobic digestion is concerned, it is commonly used technology for managing organic waste, be it your agricultural feedstock, your industrial wastewater coming from food and beverages, facilities. So any waste or any organic, any organic rich waste can be managed by anaerobic digestion process. Now in anaerobic digestion, as I said, it's a multi-step operation, but it can lead to the production of primarily biogas. And biogas, some of you those follow renewable energy sector and so on, it's one of the key energy that has been identified by almost all governments, and even Indian government actually is focusing on biogas production. But the issue with biogas is the poor methane content. It has about 50 to 60% methane content, which means that if you want to use it as a high-end fuel, for example, upgrade to CNG and use it, you have to purify it. So that is something is possible through various upgradation technologies leading to increasing the methane content. It becomes biomethane, and then you can use it as a fuel. If you do not upgrade it, that means increase the methane content, then obviously the calorific value of biogas is very poor. So obviously the efficiencies are compromised. But in addition to that, you can also reclaim water, and which is rich in nutrients, which you can use for gardening and for agriculture purposes and so on. Yeah, biogas
50-60%
is fine for it, yeah. But normally for high-end applications, as a fuel and so on. And then the third one is digestate, which can also be used as soil conditioner. Now, as I said, although this is a very old, one of the very old microbial biotechnology, almost in use for about 100 years, there are still challenges associated with this particular process. Some of the challenges are poor biogas yield, which that means the production of biogas is also is compromised. The methane content is poor. Organic matter is not completely degraded. It's not possible also. More important is slow startup and process stability. Now, to address some of these challenges, we basically thought of integrating microbial electrochemical processes into it. That's microbial electrocatalysis process. That means when I say microbial electrocatalysis, introducing conductive materials or electrodes into the system. So you can either put in electrodes in anaerobic digester or think of putting conductive material. So we followed the second approach because that's more easier and you don't have to apply any external energy. So we have been developing this technology for last few years for sludge management. Now, why sludge management? Those who follow wastewater treatment industry or applications, most of the people talk about wastewater. But when you treat wastewater using aerobic microbial technologies, and any organic matter rich wastewater is managed currently by microbial processes only, barring few industrial wastewaters which contain recalcitrant compounds. Otherwise, all wastewater is managed through microbial process, and the primary technologies that is used is aerobic microbial technology. And in aerobic respiration, as I had explained, the energy gain is quite good for microbes. When energy gain is good, they produce a lot of biomass. And here the biomass is nothing but sludge. So in wastewater, there are two types of sludges which are produced. The primary sludge after primary treatment and the secondary sludge after microbial treatment. And this sludge management is one of the biggest issue in the wastewater treatment industry. Because you have to reduce the volume of sludge if you want to manage. And the currently the technologies which are used for managing sludge are either energy intensive or costly. For example, if you want to just burn sludge, incinerate it, you have to put in a lot of electricity and so on. That is what most of the people do. Or you want to dry it and then apply a soil conditioner, then you need a lot of land area and so on. Now, to address some of those issues, and you cannot achieve any resource recovery from existing, using existing technologies. And a few speakers have spoken about circularity. In circularity, recovering resources from any kind of waste is one of the key aspects. That is something is also relevant to sustainable development and so on. So anaerobic digestion basically allows recovering some of the resources from this sludge in the form of biogas, in the form of digestion, and in the form of water. But however, it suffers because of the poor performance that I had already explained. Now, to address these issues, we have been working with the sludge management where we started with very small scale reactors, but I am not presenting that data. Now we are kind of validating the process at a few liter scale. So this is something you can see here, 15 liter scale reactors which are fed with the sludge that are coming from the sewage treatment plant on our campus, and we add conductive materials. Now we have tested different combinations and ended up with biochar and granular activated carbon. And these are our targets to produce biogas, reclaimed water and digest it. Now, I will speak about all these three products. So, in conductive material assisted anaerobic digestion, we have seen considerable improvement in biogas production. So, what you can see here data in the form of specific methane yield on the y-axis and during the operation of these reactors over a month-long period. You can see in the case of biochar, which is green, and GAC, which is orange, the specific methane yield is always better than the control reactor where you don't add conductive materials. And the methane content in biogas is almost 80% or more than that compared to the control, which is where it is around 70%. Now, in reality, 70% without conductive materials is a bit challenging. It's less than that, but because we have a bit optimized condition, that's why here you see a better methane content in the biogas. To summarize, you can see that I'll not read all data, but just cumulative biogas production. You can see in the case of conductive materials, it is much higher than the control reactor. So there is a considerable improvement in the biogas yield compared to.
I also see there is considerable error bar, especially in the case of biochar data. Any reason you have to basically?
So you mean here biogas production rate?
Biogas production
rate. Rate is quite variable actually. Because it's a long term.
Biochar case is higher than compared to
yes,
others.
yes,
So
yes.
the reason I was wondering, is there a reason for you to believe that biochar case is slightly different?
Yeah, obviously the materials are different. We have characterized these materials
based on the material.
Yes,
so
yes.
what kind of biochar you are using,
Yes.
where it is coming from,
Yes.
that affects that data.
Yes, and also there is a difference between granular activated charcoal and
now I
biochar and
understand,
so
yeah.
on. So, but the point is in if you compare the data of control with conductive materials, you can see there is a considerable improvement
That is certainly
in biogas production, cumulative bioproduction, methane production, as well as the methane content in it. So the overall, if you translate it into calorific value, this is also considerably higher and so on. Now, similarly, you get better quality reclaimed water compared to the control. Now, I will not go through all this data, but just first one where chemical oxygen demand is one of the commonly used parameter to analyze the organic matter content in wastewater. Now, so here the data is for the water that comes out of the reactors. So you can clearly see in the case of GAC, the COD removal is quite rapid, and it's almost close to not zero, obviously it's close to around 500 and so on compared to the other two reactors. In addition to that, the nutrients like nitrogen and phosphorus are not removed. Anaerobic digestion is not good at all for removing nutrients from the wastewater or any kind of sample and so on. And ideally that is required, that is good to have it in the reclaimed water, because you can then use it as a water for irrigation purposes where you need nutrients like nitrogen and phosphorus. So here also you can see the COD removal is quite good in the case of GAC. Biochar and control, here you can see it's almost on par, but the reclaimed water that you get out of this conductive material system is much higher than the controlled systems. That means the dewatering process is also much efficient in the case of conductive materials. Ultimately, you want to use this recover water and then use it for different purposes, and in all cases the nutrients remain there. So if you don't want to remove them, you can use such water for irrigation purposes. So you can also recycle the water from sludge for different applications. And finally, the digestate that is recovered is also can be used as a soil enhancer or soil conditioner. So we have tested this digestate that we obtained from these materials with the commercially used soil enhancer commonly available in nurseries and so on. For example, growing spinach, and you can see the micronutrients that we have analyzed in spinach are comparatively better in the case of control this conductive material assisted reactors. You can choose any of these parameters compared to the control and obviously the commercial product that is available. So the point here is that the digestate also has better properties, and obviously it is because of the conductive materials present in it. And these days those who follow a bit about soil science, biochar is one of the hot material these days to for carbon sequestration in soil or enhancing soil carbon pool and for a lot of other applications. So if you can combine this with this kind of processes, you can also improve the use case prospects and so on. So key takeaways, the conductive materials obviously improve the sludge digestion process through process intensification. So here, just by introducing electrochemistry, you can say in the anaerobic, conventional anaerobic digestion systems, can make a big difference as far as the overall process intensification is concerned. The second application is, how much time do I have?
You have like 15 minutes.
Yeah, then that is enough. So the second application is domestic wastewater management at point sources. Now this is important. Whatever wastewater management happens currently, it is mostly centralized and to some extent decentralized. For example, the example of decentralized wastewater management is, let's say, this IIT campus. You might have several STPs on campus.
We have one huge STP on
One, so that you can consider that as a centralized for this particular campus. Now on our campus we have, it's a small campus, Rajneesh has seen, but we have several 4 to 5 decentralized STPs. Similarly on different campuses you can have decentralized, but the decentralized capacity is still in almost more than 10 or 20 KLD capacity, and the one that you are talking about is probably in MLD and something, million liters per day. Now the one that I am talking about is further decentralization of wastewater management practice, and the reason being, now India is among world's extremely highly water stressed country, and it is going to be one of the major issues by 2050 if we don't address the fresh water use and wastewater cycling problems and so on. And one of the ways to address this problem is to try to recycle wastewater at the point source itself. And when I say point source, the source at which it is generated, it can be household also, it can be this building, it can be a resort, it can be a hotel and so on. And if you go to the rural India now, or even city areas, and in particular in Himalayan region, there is increasing trend of off-grid domestic establishments. That means people are moving out of the villages and building their houses in their farms or in small pockets, but a cluster of houses and so on. Now how do you tackle that problem? How do you manage that wastewater that is produced by clusters, small clusters or individual households or resorts and so on? If you don't manage that wastewater, it enters into the environment, it causes various environmental as well as sanitation related problems. Now there is no technology which currently can address this wastewater management at such small scale. Whatever technologies are currently available, these are just scaled down approach based on the existing technologies, but they are highly energy intensive, highly costly, and hardly serve any purpose. They just treat, they do not allow any recovery and so on. So keeping all those factors in mind, we have developed one technology which we have named as integrated hydroponics microbial electrochemical technology. Now this is currently at TRL 5, so we are at the prototype stage where we are developing prototype and going to test really at the household level and so on. So it integrates hydroponics component. I hope you know the hydroponics, and this is very popular these days to cultivate vegetables and so on, with microbial electrochemical processes in the form of electrode assembly and conductive materials. You put this concept together, and by integrating microbial electrochemistry with hydroponics using this bed matrix, plant and conductive materials, you can enable physicochemical processes like filtration, adsorption, plant nutrient uptake. So if you want to remove nutrients also, because of plants, they will take up some of the nutrients, and various microbial processes, all aerobic, anaerobic, and also the electroactive microorganisms that I spoke about. So depending upon the way you assemble your reactors and operate them, you can enable a combination of processes, which is not the case with conventional wastewater management practices. And what we are targeting here, to recover nutrients in the form of vegetables, water, of course for reuse, and because we are integrating electrodes here, and when we operate it in a microbial fuel cell mode, you can also produce some electricity. The electricity is not that high, but you can generate small amount of electricity just to light some of the LED bulbs to that area and so on. So that is what the ultimate idea is. So we have been working, as I said, with this technology for last 6-7 years. One PhD student has already graduated, who has worked on this technology. So starting with proof of concept of system design, optimization of different filler components in the reactor systems to understand influence of various process conditions. I will not go into the details of all those processes. Also testing different vegetable cultivation in these kind of processes, For example, this is the data for spinach, where you can see difference between the harvest according to the conditions. The wastewater treatment parameters, you can see more than 93% COD removal is achieved, and that is quite good data. With the conventional system, also achieving 90% is not that easy. Also, we try to understand the microbes which are involved in the overall pollutant removal process, and also the resource recovery options and so on. I'll share just the field scale data that recently we have kind of obtained, which is actually from the real field systems. So what you can see here, this data is for removal of COD, which is the as I said measure for the organic matter removal, total phosphorus and total nitrogen. So over the period of almost, so we had operated these systems for more than a year. So the data is almost for 10 months. So you can see the COD removal is quite stable over a year long period, and it is around 90%, and this is very good indicator in the wastewater treatment industry. If you can achieve stable performance over long periods of time without any interruption, and that too in the field conditions, that means under different summer, winter, as well as rainy conditions and so on. The nutrient removal, in particular nitrogen removal, is also quite good because of various microbial processes as well as plant nutrient uptake, but the phosphorus removal is hardly there because this is something is not good system for phosphorus removal. If you see here, the removal of COD is more than 90% TN, and you have certain amounts of nitrogen and phosphorus in the effluent, and this if you check for the characteristics of irrigation water, it is expected to have these levels of nutrients like more than 20, more than 5 milligrams per liter of nitrogen and phosphorus. So the effluent that we obtain from these systems is basically fit for gardening purposes. Why I am mentioning gardening? Because if this system is meant for households for managing grey water or even sewage, basically they can use this reclaimed water for gardening purposes. And this is just a picture, this is how the sewage looks like that enters into the system, and this is the water which comes out of the system after treatment, after around 11 hours or so on. This is the data for when you grow the vegetables, for example sweet basil, lettuce, and peppermint. So you get very good quality vegetables also from this one, and this is not just based on the visual observation. We of course characterize them for different nutrients. So just for examples, for sweet basil, you can see that the content of iron, potassium, and vitamin A is quite good in this the harvest that we obtain from this system compared to the harvest that you obtain from the hydroponic cultivation, where you use a synthetic medium for growing these vegetables and so on. So you can see all parameters are even sometimes better. Same for lettuce, same for spinach, and so on. So even in terms of nutrient quality, you don't compromise when you cultivate with this kind of system. Now there can be a concern of microbial contamination, but this vegetables are kind of not directly coming in contact with wastewater, and you don't know the vegetable that you are currently obtaining from market whether they are also good or not. So compared to that, basically, and anyway we wash vegetables and we use irrespective of the source from which we obtain it. So this is something is also desired here. So basically we have compared the data from our system with the existing technologies, the commercially available one, aeration based, and also competing wastewater hydroponics. You can see that the energy consumption for treating about 1 cubic meter of domestic wastewater is considerably lower than the aeration based technologies. And why this is important? Because having energy efficient systems, be it for wastewater treatment or for any other kind of application, is highly desired in sustainable development also. So here, if you are achieving this similar level of treatment and recovering different kinds of resources at same energy input or much lower energy input, this is something is highly desired. The sludge production is quite low in our system, so you don't have to worry about sludge management problem that I spoke about earlier. Currently you have to manage that because most of the infrastructure that produces sludge, but in our system the sludge production is also very minimal, so you don't have to worry about. So overall these systems we feel are very good for managing wastewater resources at point sources because they offer various advantages. I will not read all those advantages, and this is something is very much relevant to Sustainable Development Goal 6 and these 11, where it is desired that most of the resources are also managed at the point source. So this is our effort to kind of ensure water recycling at the point sources from the wastewater and so on. So the third one that I will speak about is bioproduction from carbon dioxide using microbial electrosynthesis technology. So some of the speakers have already talked about CO2 utilization using various approaches. Sebastian already spoke about the lot of catalytic applications, including electrochemical also CO2 reduction. So the process that we use is referred to as microbial electrosynthesis because it involves the use of microorganisms as catalyst, involves use of electricity as the source of energy to drive synthesis of chemicals or fuels in electrochemical systems. Now this is a typical electrochemical schematic of electrochemical setup that we use, where we use water at the anode, which serves as the source of electrons. By putting in energy, electricity, you oxidize water at the anode, leading to the production of electrons and protons. Protons they get to the cathode side at certain potential. Protons are reduced to produce hydrogen. Depending upon whatever potential you choose, either you produce hydrogen or you promote direct electron transfer based CO2 reduction process. So we are working primarily on acetate production, and here we use specific kind of microorganisms which have the metabolic pathway to convert CO2 into acetic acid. So we stop at acetic acid as far as the CO2 reduction is concerned using this kind of biocatalyst. Now how does it work? So it involves electrochemical as well as microbial processes. So in electrochemical processes, so depending upon the potential that you set at the cathode, either you produce hydrogen or you don't produce hydrogen. So normally potential higher than minus 0.4 volt, you don't produce hydrogen. This is the data for considering the standard conditions, and below the potential lower than that, you will produce a lot of hydrogen. So depending upon the potential that you have at the cathode, either you will produce a lot of hydrogen or you will not have hydrogen production. Now depending upon what one wants to achieve, you can choose the microorganisms and the process accordingly. So for example, if you want to convert CO2 to acetate, this is the reduction potential that this is theoretical. In reality, you don't achieve that reduction process at this particular potential because of a lot of electrochemical losses and so on. Similarly, this is the potential for the methane if you opt for direct electron transfer based process. Now if you use microbial catalyst and for example opt for the direct electron transfer based processes, it is feasible, but the yields are quite poor. So normally we go for hydrogen based production. That means where you first produce hydrogen, first means simultaneously in the reactor, and this hydrogen and CO2 are then used by microorganisms to produce acetate. So these are some of the examples of microorganisms that we routinely use in our systems. Now here one point that I would like to mention is, so those who have heard about gas fermentation process, so fermentation is common process, but it's sugar fermentation or other kinds of glucose fermentations and so on. Gas fermentation is one of the emerging technologies now to also to utilize CO2 or different other kinds of gaseous products like nitrogen and so on. So this microbial electrosynthesis, in other words, is a combination of water electrolysis with gas fermentation, but both enabled in single reactor. So you avoid using two different reactors, you avoid using highly pressurized reactor conditions, which is prerequisite for gas fermentation processes, by just having in all those conditions in one system. So we have been doing this work for some time now. So we have also worked with industrial gases directly. For example, brewery CO2, I think you mentioned today morning that you don't have to purify it because CO2 is present in high concentration. Even if some impurities are present in industrial gases, microbes can deal with them. Microbes do not get easily affected, unlike for example maybe metallic catalyst, which can easily get poisoned because of traces of impurities and so on. But this is not the case with microbes. So we have been working with biogas, brewery CO2 and some other industrial gases, where this is just data how it looks like when you monitor acetate gas production and so on. So now we are at the level where we are testing these processes at a bit higher scale. So at 20 liter scale reactors, we refer these reactors as electrobioreactors. combination of, as I said, gas fermentation or fermenters with electrochemistry, which is funded by BIRAC. So we are now testing this process at bit higher scale.
Typical outcome from a 20-liter reactor, like.
So this is currently ongoing. So it's not optimized. So
But
we
an unoptimized system, what do you?
I mean, we have set it up a few months ago, and we are still struggling with
You must
there's-
have an idea, no? That
So-
theoretical calculation that this could be an outcome from. A
see, our target is to achieve at least 20 grams per liter.
20
Constant,
per
tighter,
liter.
tighter, yeah.
Okay.
And so we don't worry too much about Faradic efficiencies, because I was explaining to
That's
him
still
also.
20 liters, 20 gram per liter is like 400 grams, so it's pretty good number.
Yes, that's our target. And Faradic efficiencies are always more than 80%. Specificity is also not issue, because these microbes produce only acetic acid. Yeah. Yes, a little bit of formic acid is there because it is intermediate in the metabolic pathway, but it is produced transiently and then it is consumed again.
It's a batch process.
This is a batch process, but we want to,
Keep in mind, the batch process, I mean, always favorable for acetic acid. The moment you change,
continuous. Yeah. No, so at the lab scale reactors, a few liter scale like 1, 2, we have operated in continuous mode also, and it produces
Bioprocesses
continuously.
are basically known to be working as batch processes, and large scale production are actually
Yeah.
better outcome.
Even metal based on
More, okay, better. Now, in our cases, we have operated these reactors even in continuous modes, even for a year actually.
See, large
And it
scale
is- it
operations
produce-
for polylactic acid, lactic acid production were all batch processes.
Yeah, mostly batch process. Yes.
and I have one point you have acetic acid and then it can be slightly acidic.
Yeah, with accumulation, it starts becoming acidic.
Then kinetics in favorable for the
Yes, yes, yeah. So what we do normally, we use buffered medium, high strength buffered medium, which adds to the cost, but there is no other option. The other option is to recover it continuously, extract it continuously. So that is something.
acidic medium hydrogen evolution dominates.
Yes, yeah. So that's what we see. Eventually, a lot of hydrogen coming out of the system. With time, if the pH drops, considerably below 6 and so on, when it completely becomes, acidic and so on. But we normally tend to avoid that by having high strength, buffer medium in our case. So this system we also use for biogas upgradation. For example, we feed the biogas to the cathode side where CO2 fraction of the biogas gets reduced and methane fraction gets, enhanced. So for example, from about 58%, we have achieved more than 90% methane content. So this is one of the interesting approach even for biogas upgradation processes. Faradic efficiencies are also higher, and this is achieved at considerably lower cell voltage, about just 2.6 and so on. So these are the processes which are currently at TRL 3 to 4 level, and we are trying to kind of scale it up further and assess whether we can reproduce the similar kind of performance at the higher scale and so on. One important aspect as far as the role of microbes in these processes are concerned, here you can see the cyclic voltammetry data. So the red and black traces are for the cyclic, are for the cathodes before microbial inoculation and immediate after inoculation. You have the hydrogen evolution potential somewhere, here we can say broadly, and the reduction current is also quite low. But when microorganisms grow at the electrode at the end of the experiment after a few days or weeks, when microbes form biofilm on the electrode, now this is referred to as biocathode, you can see the hydrogen evolution potential is also considerably lowered and the reduction current is also considerably increased. So they play a very important role also in enhancing hydrogen evolution catalysis. In other words, it means that the overall energy input to the process is minimized considerably, because you have to put in a lot of energy to split water and produce hydrogen. But microbes, because they have the hydrogenase enzyme in their system, they can also play important role in basically. Sorry, we haven't done that, but I think the impact should be more or less the similar. Yeah. So and this is something we see consistently in all electrosynthesis experiments. So they with time they also bring down the overall energy input down. That in terms of efficiencies, that is good, and in terms of cost, that is desired. So at lower energy input, you get this one. Now acetic acid, of course, is platform chemical, highly desired, but you have to keep in mind that it has a still low value. And another issue is that you cannot compete with the existing chemical catalysis process, Monsanto and so on, very efficient, and but of course they are not sustainable processes. So what we have been also working on to add more value to the products that we produce from carbon dioxide, and one of the processes that we are working on is yeast cultivation. So Saccharomyces cerevisiae, brewer's yeast or baker's yeast is something you have heard about. Now we use this yeast, genetically modified as well as wild type, to produce some terpenoids like sclareol, beta carotene. Sclareol is a high value product used in perfumes. Beta carotene is a pigment, is also widely used, and also just yeast biomass as protein source, or just as a yeast extract, which is commonly used in all research labs or industries all over the world in microbial growth media.
These are the material which you are showing is basically the biomass which is produced by the organism.
Yes.
It's not a reaction which happens in presence of this.
No, CO2 is used as a carbon source.
But then
So here for yeast we are not using CO2. We whatever acetate is produced in in the first step, this is a two-step bioprocess now. So acetic acid is produced first, and that is fed to yeast. Now yeast cultivation is primarily done with glucose everywhere, and glucose comes from energy intensive process and so on. So we are trying to now produce all those products or bit high value products from acetate that we produce from CO2. And overall basically it makes sense to do that, for example for biomass and also for sclareol. So we had done some preliminary economic analysis, and here
even the previous one, if you are going from CO2 to acetate, that's one way.
Yeah.
Let's say you have methane.
Yeah. Yes, methane.
That itself is
Yes,
a
yes.
lot more value addition.
Yes.
So it may not be CO2 utilization, but it's like gas to liquid, gas to solid kind of.
It's already happening. Yeah.
A lot of these things are basically.
Yes,
but,
yes.
Is it easier to control the size of the molecule through this route?
Yes. So methane is
For
already.
example, he talked about aviation fuel.
Yeah.
Now, if it may not have that sustainable word in before, if you don't use CO2, but if you use methane to convert that into aviation fuel, that's
the sustainable name actually. Now it's actually synthetic aviation.
yeah. So synthetic, let's
Mhm.
say. So, is it
No, actually it's
doable, or how easily it is controllable? That's the main point.
So with with all gaseous feedstocks, of course, it's not that easy, because in particular, if you talk about hydrogen,
Not hydrogen, I'm saying
methane is reasonably soluble in
hydrogen
water.
is very expensive. but
Yeah.
Methane is much
Yeah, so
cheaper.
meth-
If
with-
one can do that, that
yeah.
is going to be really a game
Yes,
changer.
yes.
So, Rajneesh, what you're telling absolutely right.
Yeah.
Yes.
We don't need big molecules.
Yeah.
First, we need methane.
Methane.
From 6% to 15%, we want to increase the target by 2030. Our
Yes.
gas penetration should be from current maybe 5% to 15%. That is huge number. We are putting city gas distribution network everywhere. So the first molecule is methane.
Mhm.
At least 5%
So-
energy problem.
So
There's
even
lot
in
more value addition.
yeah,
You
even
can
in
do it with-
bio-based processes, methane is already considered as one of the feedstock. And methanotropes are the group of microorganisms that can oxidize methane.
So
And
it
you
is a
can produce.
market.
Yeah.
It's a fuel market. Methane, if it is a food market.
Yeah, singles. But the methane-based bioproduction is already.
Anaerobic digestion. There is around 40% is CO2 is coming.
Yeah.
Now we all are struggling what to do with the CO2. This is pure CO2. Can that CO2 can be back into the methane? So
Yes.
we get more methane.
Yes, that's possible. That process is referred to as electromethanogenesis. I haven't spoken about that, but you can convert that CO2 to methane using methanogens and increase the methane volume overall. Yeah, that is something.
Then
Yes.
existing infrastructure.
Yes.
And
So
because this biogas plant had its own carbon footprint.
yes.
We not recycle that.
So in the beginning, I had mentioned two approaches, either electrode-based or conductive materials. We are following the second one because it is cheaper. Now, electrode-based means you have to put up additional electrolysis system, where you have to feed that biogas like I had shown here in this case. Now, this is increasing the methane content in biogas that you are referring to. But here you need additional reactor, that means additional cost and so on. So in the first,
need to have a secondary reactor to feed.
yes, yeah.
But
But-
you have to separate out and then you have to put.
You can feed the biogas directly. You don't have to separate CO2.
Okay, so the biogas means methane along with the
CO2, yes.
In this mixture,
Yes.
CO2 will be
You can
converted
keep
into
enriching
Yes.
methane.
Yes. You will just methane will not be used because you will not be using
Competition, right?
you will not be using methane utilizing microbes here. You will be using methane producing microbes. Yeah. Now, why we are also integrating conductive materials, because you can do that in one reactor only. There you will not obviously increase the methane content more than 90%, but considerably higher than the normal scenario. So you can still achieve better use case prospects for that process and so on. But that is also possible, electron. Yeah, that's all. Yeah, so this process is feasible, economically. So we have been developing now a bit higher scale process for yeast production, and this is what you can see. This is first test, and then this is how it looks like when you dry it. And we have done some calculations. So this process basically allows yeast biomass production with around 2.6 times lower CO2 emissions and at 1.2 times lower cost than conventional glucose. So this is the project which we have waiting for the funding from DST, where we want to kind of scale up this process and test whether
No one has got the funding yet.
I think 2-3 people have got it, but all right. So I will skip the summary. Basically, I hope you got something out of the presentation, that you can try to explore such combinations. This is a unique combination, microbes with electrochemistry, and do a lot of things with the conventional processes, as well as try to develop some new processes. With this, I thank the funding agencies in my research group, and I thank again Professor Rajneesh for the invitation.